So there we go. So hi, everyone. Yeah, we need to do introductions. So Tishy, Will, and I'll kind of introduce ourselves briefly, go through a couple of just high slides, and then I don't even know if we want to do a breakout room. If it stays this small, we'll all just introduce ourselves here. The last session we had 30-ish, and so it was better to go into breakout rooms to do the introductions. Hopefully it won't take too long to get through all of that. Yeah, and then we'll get started. So yeah, I'm Josh Moore. Hi, from the University of Dundee. Open microscopy environment. I know most of you, or we've met at some point. In a previous life, I was responsible for building Omero, so a data management platform. I know Jamie, so I'm not going to restart. Yeah, have since been trying to get this whole NGFF new file format concept going. And yeah, let's see how that works out. Just doing introduction, Shilpa, so you haven't really missed anything. Christian, Will, let's go next.
Okay, yeah, so I'm Christian Tischer, nickname Tischi. So I work at EMBL Heidelberg in the light microscopy facility, but now Center for Bioimage Analysis. And the reason I'm in this workshop is that for the last 1-2 years, I have been quite a lot into sharing big image data via object stores, which is what this workshop will be basically about. So yes, that's why I'm here. Okay, then Will.
Hi, I'm Will. I work on the OME team, mostly on the Emero server up to date, and on the web side of the Emero server work. My involvement with the NGFF stuff has mostly been about how to display, visualize the NGFF data that we're generating in various clients.
I mean, Josh, if we do it all together, then we can also do it now, and then we have the high-level slides afterwards, or? Okay.
Yeah, let's see, we're up to 23, but if we keep it short, then no one will get too bored. So, I mean, we have the spreadsheet. Actually, I guess I can go ahead and share that. So that I sent around in the emails these documents. So share that. You're welcome to tell us anything you'd like to about yourself, and the spreadsheet that's linked from that document. It's also useful to have, oh, yeah, that's the spreadsheet. It's useful to have that information also afterwards. So tell us whatever you'd like. Other than those documents, let's use the Slack share. If you'd like to say anything, let's stick to that share. And then I'll go through, I guess, my screen to do introductions. Yeah, so what you want to get out of this, maybe. Nico, you're at the top of mine.
I'm at the top of yours. Do I see the spreadsheet? I see the spreadsheet somewhere. I mean, I would just...
Through the Zoom.
Yeah. Sorry?
I'm going through the Zoom, so
okay.
Just introduce yourself.
So I was hired as a bioimage analysis specialist at the microscopy facility in Basel, like almost 10 years ago. And this is what I did for the first 5 years. And then it turned more into like a data shuffling around specialist, chief technology officer, something, image analysis back office consultant. So which means I basically shifted off all of the real life user projects, problems, whatever you want to call it, to my colleagues. And I'm mostly, I mean, I'm not doing that many image analysis things anymore myself. I help them when I can, but mostly I'm dealing with running our Omero, which we also almost have since a decade. I mean, it's more than 8 years. I checked this morning, we have roughly 190 terabyte of raw data in there, and there is, I have to point that out, especially for Tischi, we don't have any screening data in there. So this is mostly confocal, spinning, disk, white field, stuff like that. You know, the daily bread and butter type of microscopy images. So plus the whole infrastructure around that, we're producing a lot of data, and people are struggling with getting it around, accessing it, processing it, whatsoever. We run a huge virtualization environment that currently also no one else is left managing, which means I'm doing that. providing our users with a desktop infrastructure where they can access their files with Fiji, with Elastic, with whatever they want, with Conda stuff, with MATLAB, with proprietary things, Imaris and that stuff. Yeah, we're operating a deconvolution server. I wrote the connector to Omero to push files back and forth from that. We operate a service getting data more or less automatically off the microscope systems to our virtualization infrastructure. And in my spare time, I'm doing, there's none left.
Yeah, there's no time left. Blair?
Mute button. Hi, I'm Blair Rossetti. I work at Genelia in the Advanced Imaging Center. I am nominally supposed to work with our visitors on their image analysis problems, but during COVID times, I have switched to working on infrastructure stuff until we have visitors again.
Nicholas?
Hi, I am Niklas. I am a master's student from the University of Marburg and study physics. And I work in the group of Knut Drescher. And we are doing research with biofilms, mainly Vibrio cholera. And I'm just interested in learning new techniques for image analysis. That's why I'm here.
Great, thanks. Eric?
I'm Eric Troutman. I work at Genelia in a software engineering group that supports various efforts at Genelia. For the past 5 years, I've been mostly focused on software for large-scale EM reconstruction efforts that's based on software developed in the Saalfeld lab. So I work closely with Saalfeld and Privish, and I'm interested in hearing what others here are doing.
Cool. Arturo? Arturo, do you want to introduce yourself?
Hi, sorry. I'm Arturo Pimentel from the National University of Mexico. I'm academic technician in the National Laboratory of Microscopy. I'm here. I'm very glad to be here, and I hope a very nice experience. Thank you.
Bengt?
So I'm Bengt from George Mason University, and I'm the technical manager of neuromorph.org, and I'm interested in various different formats that we receive and manage, and how we could facilitate for maybe future different formats that we may receive.
Always good to be ready. Kevin?
Hello, I'm Kevin. I'm a postdoc in Basel, and I enjoy building image processing tools for microscopy. Recently, I've been working on things related to calcium imaging, cell segmentation of light sheet data, things like that. And I'm excited to be here to learn about new ways to learn and share large data sets.
Thanks, Kevin. Jamie? No, Martin was first, then Jamie.
Hi, I'm Martin. I work at the Francis Crick Institute in the electron microscopy core facility. Most of what I do at the moment is some hardware development for the microscopes, and then a lot of software development for handling our big data, segmentation, visualization, and so on.
Mm-hmm.
I'm Jamie Sherman. I work at the Allen Institute for Cell Science. Kind of a hybrid role as in applied. My background's in applied math and biotech, but I've done a lot of software development, and so I'm leading the efforts here on image reading and metadata translation, et cetera, at least from the practical doing side. And so, yeah, I'm interested here in, we've put together some approaches to tackle some of these problems, but I'm really interested in seeing how other bright people might have done this better. So yeah. Anyway, that's my interest. Cool.
Thanks, Jamie. Shilpa?
Hi, I'm Shilpa. I'm from Icahn School of Medicine at Mount Sinai, New York. I work as a senior scientist here and helping biologists analyze and quantify their data. So working on a lot of microscopes even before, like a lab-built light sheet microscope, we used to have like high throughput, high resolution data, which would take up terabytes at a few seconds. So this is really just to address that problem and how generally we could improve our storage and processing of large data. So yeah, excited to be here.
Geronimo or Geronimo?
Hi, I'm Geronimo. I'm a postdoc in Munich. And we're going to generate some light sheet data, and as part of the proposal that is funding my postdoc, we said we were going to share it publicly, and I would like to learn how to do it properly or in the best way.
Hi, I'm Matt. I work at UW-Madison in the Cryo-EM Research Center. I'm a system administrator, but I also do software development. Most recently, one of our projects is on correlative light microscopy, and we're trying to make a Java application that would align MRC images.
Hello, I'm Florence. I'm a postdoc in London, and I'm doing a lot of microscopy, and in particular, I'm using light sheet microscope. And so I guess I have the same kind of interest that Shilpa had about like big data, how to handle these and storage and everything.
Hi, I'm Anna. I'm working at St. Jude Children's Research Hospital at the Center for Bioimage Informatics, and we develop image analysis pipelines for biologists. And among other things, I'm working with large data sets. This is why I'm here.
Okay, thanks. Christina?
Hi, my name is Christina Bergaki. I'm a senior support specialist at the Rockefeller University. My background is in biology. I did a postdoc in neuroscience, and I became a microscopist by trade, I guess. And now we're trying to like sort of up our image analysis and processing game at the core at the bioimaging facility. And we have a light sheet that generates fairly large data, and like we're just kind of trying to figure out how to handle better the sharing and data processing among the users and all that stuff. So that's why I'm here. We're just trying to figure out how to up our game.
Sorry, I was muted. Is it Mariamba? Oh, hold on, you're muted.
Good morning, everyone. I am Mariamba. I am from the University of Nevada in Reno. I am an assistant professor. My background is in molecular biology, focusing in finding biomarker for asthma. I recently generated a whole long slice images that are big data, and I am not sure how to analyze them. I am interested in finding the inflammatory cells and fibrosis, but I am not sure how to analyze it. That's why I'm here, and I've been here for 3 days now, and I am really excited. The coding part, I don't even know what's going on, but I was just excited to see how all that was done. Thank you.
That's great. Glad you're here. And finally, David.
Yes, hello everyone. So I'm David from France, Nantes in France. I'm a computer scientist working in image analysis and working mostly with microscopists, but we have also some concerns with mass spectroscopy images, and we have questions on how to merge the information coming from the different modalities. So question of formats are interesting for us. Thanks.
Okay, great. If we missed anyone, feel free to speak up. In general, feel free to holler at any time. So our experience from the previous workshop and what we've heard from other people giving workshops is that after, right about 3 hours, everyone starts to kind of nosedive. It'll start getting late. We'll all get tired. So we're going to do, Tishy, if you want to go ahead and share acknowledgements now, because obviously this isn't just our work. This is, coming from lots of people, and it would be sad if it got missed at the end. So I'll give the floor to Tishy.
Yes, okay, thanks for the floor. Oops. Okay, so few acknowledgements of some people: Konstantin, David, Sebastian, Joan, Draga, Nicolas, Stefan, Tobias, Trevor, and Ilan, and many, many more. So the ones that are listed, if you look at the names, what they do, so you see Big Data Viewer, N5, NZAR, OME SAR. So essentially, really what we present today is kind of a community effort. And I think what the OME people are building on is stuff that kind of maybe existed already in some way, and now we are trying to standardize it and so on. And this is a big discussion with the community, including you if you want. Yeah. So we will show several things today, and they look like they are, but that would be the wrong message. I just want to say now, this can be all still changed, right? So this is just a proposal. So if you don't like anything about it, this is a community discussion, okay? So that's very important. You want to add something, Josh or Will? Okay. Then one slide, because maybe since you are interested in image data, so the Eurobioimaging at EMBL Heidelberg, they are actually kind of desperately looking for someone that could coordinate image data services across Europe, which would also could or should, in our opinion, involve stuff like what you will learn today. So basically teaching like all the facilities, best practices of how to handle all the image data. So if you think that's interesting, and you also think like you like to, coordinate people and also talk with them and so on, then think about applying there, yeah? Okay. So very good. Any questions so far? If not, I think I can go on, right? Because the first session will be actually like a motivational session of why we do what we do. So I will talk about challenges of browsing and sharing large 3D image data sets, because I have been through this, as I said, in the last 2 years, and actually found some solutions, I think, that we want to share with you. So maybe first, let's rehearse our very short and concise title. Towards a community standard cloud compatible next generation brackets NGFF file format for bioimage data, okay? So what I, in my talk, want to talk about the fact that we're saying there should be a next generation community standard, which I think raises the question, what's the current community standard? Okay, and why do we need a new one, right? So I think, why doing all of this work? So one may have different opinions on that, but for me, working like for a long time now already in a light microscopy facility, long before people even knew what Python and Hapari is, this is, I think, what I would say is the community standard, at least from my point of view, right? So you have a TIFF image somewhere and you drag and drop it onto Fiji, and then you look at it with the hyperstack viewer. So from where I come from, this is the standard. Okay, so, and I will compare everything against this. Obviously, there's other solutions, right? But I would not say they're yet a community standard. Good. So when did this standard stop working for me? It stopped working for me when I got involved in this project, which is a very big collaborative project where we actually made a mapping of light microscopy gene expression data into a whole body electron volume of electron microscopy volume of a Platina reyes animal, which is an animal that's interesting for developmental biology. And as you will see, this had several features that broke kind of the current standard in a way. So first of all, so at the heart of this project was a big EM volume actually acquired in Basel. So as you can see here, it's about 300 micrometer along one dimension. Oops. 10 by 10 by 25 nanometer cube, which amounts to several terabytes if you look at how big it is. And let's now say you save this as an OME TIFF file. What you will do is you will save many, internally of this file, you will have many planes that are saved as these TIFF planes. This is just how TIFF works. So in fact, that's actually still okay, because in ImageJ, as most of you maybe know, is you can say open TIFF as virtual stack. So what then will happen is the software will not open all these 8 terabytes, but if down here you move along the Z axis, it will just go back to this file and fetch on the fly from disk for you the plane that you currently look at. So that means this is working, okay? So there we wouldn't need a new solution because this has solved this problem, yeah? So you can easily look through all this beautiful data. This is, by the way, the plateau rays, some muscles. This is his brain, kind of. These are all the brain cells, so it's really a beautiful data set. But what happens now if you want to look at this plane? Which is what we really want to do, because the animal actually, it was imaged in a very weird angle. We couldn't really control how it was sitting on the microscope. So we really want to see this axis. This is the biologically most meaningful plane, yeah, not those guys. And then what do you do? Okay, so first of all, in the ImageJ hyperstack viewer, which I said is, I would say, a standard, you can't even tell it that you want to see this plane. And even if you could, if you then would ask the OME TIFF file format to give you this information, it would have to load all these planes just to fetch like 1 pixel from each plane. So that means you have to load like the whole 8 terabyte into RAM in the worst case, although you just want to see 1 plane. Yeah. So this kind of arbitrarily slicing, I think, is both file format wise and on the viewer side not solved by a standard solution, let's say. Then the next issue we had is, so we have this EM data set, which is really high res. But then we have also many light microscopy data sets, which we actually registered into the EM data set. And these light microscopy data sets, they are indicating where certain genes expressed in this animal. So for example, here you can see this is spaces where we think the myosin protein is expressed. Now, you can load these individual planes after registration. So that's, as I said, no problem. But then they have a different amount of pixels, right? So this guy has only 500 by 500, whereas this is much bigger. So if you want to see them on top of each other, just in good old ImageJ, that's not possible because the ImageJ viewer wants 2 images that have the same number of pixels. So that means either you have to upsample this guy or downsample this guy, which is both not good, because if you downsample this, you lose resolution. If you upsample this, you increase your data size artificially, which is also not good. So also for this, the standard solution doesn't really work. And then finally, so this is a really rich data set, actually. You can explore this probably, for a long time. And I think there's a lot of interesting biology to be discovered that we don't even think about. So we really want to share this as a resource with the community. So we were really thinking, how can we now share all of this beautiful data? And we had tried several things, but one basic issue is if you just put it on a file server at your institute, your IT department, is what I learned, they don't like the idea of opening this up to the public. So they just say, no, we don't do it. And even if they would do it like this, it's the question what you would even give the people. If you give people just a link to that file, would that mean they always have to download a terabyte if they want to look at it and stuff like that? So this is essentially also a challenge, I think. All right. But since you're in this workshop, you will learn the solution to all of this, and it's working today. So And then you can share all this data. People can like arbitrarily slice through it and overlay the different scales and look all of this in the cloud kind of. Okay, so this is kind of the outlook, what we will walk you through now, how to achieve this and what could be best practice there. All right, so this was my little motivational intro. Do you have any comments and questions maybe already? Someone?
So are you, question here, so essentially it sounds like with your transformation, like wanting to read off image access and stuff like that, that you're hoping to leverage Dask and things like that for mathematical transforms on the data, or is that the gist of it?
Yeah, I mean, I think the solution we will propose on the Python side will involve Dask, let's say. Yes. Yeah.
Okay.
So that's correct. Yeah.
Thanks.
Yeah, and if anyone has an idea for Dask in Java, we're all ears. So.
Any other comments? Good, not then. I stop sharing. And I think, yes.
Okay, so this is kind of the other half of the story, which is trying to get all this stuff into the cloud, and we'll touch on all of that. So, take this long title, which we're aware of, sorry. I'm going to focus primarily on just the getting it into the cloud and getting it back out of the cloud part of the story. You may have seen on Twitter and in the Slack room, so someone commented this during the first session this morning that I didn't bring up the word fair. So for what it's worth, this is the A and the I of making your data fair. So accessible and interoperable. Findable is a different part of the story, and reproducible is yet another part of the story. So we're not talking about everything here. It's really, trying to keep things simple just for the purposes of getting us out of here in 3 to 4 hours. The other thing that already got mentioned in the Slack channel, so if what we're working on here is what's a format that works well in the cloud, why yet another format? I will do my best to defend ourselves against the XKCD challenge. It's a valid one. It's one we should always be thinking about. I think we have reasons for doing what we're doing. So you can be the judge of that by the end of this. So take a step back. Here, I'll just go ahead and create all this. To date, OME has one format that we support, as Tischer mentioned. There's one current standard, and that's OME TIFF. Its limitations. So first of all, TIFF is really well known. It's almost all the, it's some huge percentage of the data that we actually see on a regular basis. Like checking the IDR, the image data resource, it's like 99%. So, there's clearly a barrier here when it comes to replacing it as the standard. It does have big limitations though. So this 2D-ness causes a lot of problems. And we worked to fix those previously, at least for some domains. We went through this process. Sebastian Besson led this effort of building these pyramids, these multiple resolutions into OME TIFF so that you could do this zooming, Google Maps style. What you do is you downsample the image, you store it all in the same file, and then if you want to zoom all the way out of a very large image, you no longer need to download 350 gigabytes. You're only downloading something like 20 megabytes. Only loading that much. Unfortunately, next slide. Things are slow on my side. What that means is you now have a bigger file that you upload to the web, and someone has to download it, right? I'll leave you guys to handle whatever's happening in Slack. So this is the problem that Tischer, if you go through his story, he gets to the end where something's working, but not if you have to upload it to the web. So the solution to that, the basic solution to that, and we'll certainly be diving into it more, is to make your file smaller, right? So what you want to do, things are really slow, is take one monolithic file, cut it up into many pieces, and put those up, in the cloud. We'll talk more about exactly what the cloud means. It's a pretty inspecific concept, and we need to define clearly what we're all talking about, make sure we're talking about the same thing. By doing that, so by having lots of small individual objects that are in the cloud, you no longer need to put a server in front of your data. So we build Omero. Omero is basically there to have something, it could also be called in the cloud, that lets you download bits of data via an API. But IT department must run that server. They must provide it to the world, and don't want to do that. It's something else you need to take care of. So this change in paradigm is to say, well, okay, we're not going to have a server that's doing all this. The data is just going to be available in a format for everyone to access, right? So each of these links you can think of as a URL that's sitting out on the web, and anyone can start accessing them. You know, so if you're thinking about what your users want to do, they're in the browser, they're all the way zoomed out, you know, in Google Maps, so that they're only loading, you know, they're not even loading 21 megabytes, they're loading pieces of 21 megabytes as quickly as they'd like. For this particular data set, like I said, the data set is in total 350 gigabytes. That's over 900,000 pixels wide by almost 400,000 pixels tall, right? It's 2D, but it's still quite gargantuan, at least, especially for the days when we first got it. It was, for a while, it was on Wikipedia, the largest image on the internet. And so the chunk size we've chosen is 1 megabyte. And it's something that others have done. Amaris does this. Other software that's much older than what we're talking about here, you know, basically says that's about what you can get on the fly. So you pick a good chunk size for what you want your users to do. And then as they zoom in, you know, they're not loading all of the 1 megabyte chunks from the large data. They're now looking at just a region. And then you kind of have what you want. So that's the overall goal that we're trying to get to here, right? So then it comes down to what's available to let us do this, right? So TIFF, I've talked about. HDF5 ticks a lot of the boxes, and maybe for some of your use cases, actually using HDF5 makes sense. So we're not trying to say that any of these things are bad or going to disappear, but there comes a point at which things are too big, and that's when you really need to move to this web scale. So the two formats that we've been looking at are ZAR and N5. They developed in separate communities. They're very, very similar. They're very, very simple. ZAR developed in the Python community, N5 in the Java community, but they both work exactly the same way. You have lots of little files, and you can attach metadata to what you're working on. So let's go through the table, look at kind of some of the pros and cons, because that's what we'll be focusing on. So TIFF, once we added the multi-resolutions to it, is a viable format. This is actually part of the TIFF spec, so we haven't created anything new here. We're just making it available to users. With HDF5 and ZAR N5, you can store multi-resolution data, but there's no specification. So here's where I'm defending myself from the XKCD comic that, you know, just the fact that you can write data down with a certain library, with these ZAR N5 libraries, or even HDF5, doesn't mean the community has really agreed on how they're going to write it down. So what we're basically here to do is, you know, take the community and say, how is it that we're all going to write this data down? Let's agree on it, and then all of our tools can start using it. And so that's what we'll be showing you, is things that have been agreed on to date, and then maybe we'll get into things we need to agree on moving forward to make this more useful for all of you. One of the big jumps then, as we mentioned, TIFF is 2D, critical to be 3D or higher dimensional data. You know, certainly, you know, as you start rotating your light sheet data, is that, do you count the rotation as a dimension? Is that a fourth dimension? Do you have a time series? You know, 6, 7D data is certainly possible. At the moment, we're working with 5D data. That's where we've kind of drawn the line, but it'll keep growing as new use cases arrive. And then critically, TIFF is really slow kind of across the board. It's not, I think TIFF was, you know, the current version of the spec came out late 80s, I think, and hasn't been updated since. So, I mean, you can kind of imagine it's not going to be built for the use cases we're working on. HDF is much faster, but it's still slow to write in parallel. So this is one of the big problems that many people, including, you know, Tischer and Amaris and others, have worked on, is can we find a way to, if we're working with HDF5, to do it faster? For example, Amaris itself recently released an Amaris writer that is faster. We were just talking about that earlier. You know, so if you're using HDF5, you know, there are solutions so that you don't need to go all the way into these cloud formats. And then finally, so with these small formats, when you can start to address individual bits, the proposition is that reading and writing is fast. A caveat there is what that means is you have lots of files and you have to get them into the web somehow. So you have to upload them, and that's a costly process. So we'll look at that a little bit. But before you go to your IT department and say, I create 10 million files for my 10 terabyte data set, you'll need to be aware they'll probably push back. And so you'll have to have a conversation with them. So there are things that are still problematic. The one that we're trying to make easy here is the users accessing the data. You still have to do work to get the data public, right? So don't forget that. It's not a complete panacea. Okay, this is the last bit. This is just kind of the overall OME vision slide. So if you've never heard about OME before, it's basically our business to try to make your data as reusable as possible. That's what we focus on, as opposed to analysis or anything else. That's our business. For a long time, the way we did that was this library called Bioformats, which is basically the translation library for converting from hundreds of versions of data into a common specification. It's there, you can use it. It's a great library for what it does, but basically we're telling the community this isn't working anymore. So as your data is growing, converting, using a library to read your data every time you want to access it's just not making sense. Basically, you're producing too much CO2 just to see your data. And we really, really want this to stop. So there needs to be a common format. We really want everyone to start converting their data into a small number of community-supported open formats. So that's what we're talking about here. The only thing that you then start to realize is then, some people have taken this on board. These, Janelia and Dresden have worked on formats like BDV and KLB. There are the N5 and ZAR formats that I mentioned. It starts to become that you have more and more open formats working with more and more tools. So that's a problem that we need to be aware of. So as a community, we need to make these agreements and shrink what we're talking about. So that's the process we're working on here. What we've done from our side is go through a number of specifications. I won't talk about them here. So those are the details we're about to get into. All of that is happening online. So these are the primary links if you want to look at them. The specification itself lives here under ngff-openmicroscopy.org. It's in progress, so we're constantly updating it. Suggestions welcome. The conversations about what's happening to the specification happen on ImageSC. So follow the tag OME NGFF if you want to hear more. All these slides are a little bit higher in the Slack. You can click on the links if you want. Yeah, that's it from my side. Any thoughts or questions that anyone wants to get off their chest now? While Will's getting set up.
Josh, just curious, is Z5 still alive?
So Z5, for everyone's benefit, is a library that's written, well, initially it's written in C++ and then wrapped with Python. It lets you access both ZAR and N5 at the same, with the same code. It's a translation library itself. As far as I know, Konstantin's still supporting it. It's there. For anyone, so not to do Konstantin any disjustice, but if anyone's looking for a C++ and Python library, though, you may also want to look at TensorStore from Google. So basically, we need to do some benchmarking and figure out the fastest way to access these things. The ecosystem is developing pretty quickly in the background, but I take that as a positive, even if it means you kind of need to be aware of what's going on. There are people who are interested in building good libraries. So I think that means good things for all of your data if you start to work in this direction. So saying that to everyone, not just to you, Blair. Anyone else? Okay, go for it, Will.
Yes, a little bit late, but okay. It's David speaking. I have a question about dimensions of images. For the moment, the dimensionalities of the data are mostly spatial dimensionalities? And are there any possibilities to manage the time dimensionality, for example, or spectral dimensionalities? Or is it so in this format? Or do you think you will discuss this later? I'm mostly interested in the hyperspectral images when each pixel is a spectrum. And in that case, the question of upsampling or downsampling, maybe we may want to manage differently the dimensions. So
yeah, so at the moment, the dimensions are the 3 spatial dimensions, time, and multiple channels.
Mm-hmm.
That's coming from the current OME TIFF spec. So adding in new dimensions is something we'll just have to discuss with the community. So I don't think there's anything in the libraries that would prevent it from happening. It's mostly about making sure that all of the viewers know what to do with the data when they receive it, right? So in some sense, you need to also specify what it's going to look like, what the user can expect. I don't exactly know when that'll happen. So to some degree, if you have example data and you want to get involved, I think that would be a great thing to move forward.
Okay, thanks.
Yeah, cheers. Anyone else? Okay.
Right. Okay. Let's see. So that the link I posted in there is to this page that I'm going to walk through most of the stuff on this page. There's a Jupyter notebook that I'm going to go through here. If anyone's gone through this, these install steps to install this environment, and you've either checked out this repository or you've downloaded this the notebook file itself, you should be able to run this notebook locally. Or if you want to follow through without installing it, everything, you can click on this binder link at the top here. It should start a Jupyter notebook for you, and then you want to click on the OME-ZAR intro notebook. So I've already started this up, and yeah, you can follow through here. So you only need to run this yourself if you want to actually play with these code samples. But first, I'm going to start just for those not familiar with ZAR, just to give a little bit of an overview of the ZAR format, and then give you an idea of how we've implemented that to store images in the OME-ZAR spec. As Tischer said, this is a kind of an evolving spec, but I'll give you an idea of what it looks like at the moment anyway. And okay, so first sentence here is just from the ZAR documentation. ZAR is a format for storage of chunked, compressed, n-dimensional arrays. And then you can also arrange these arrays in a hierarchy with additional metadata in what's called groups to encode the kind of data you need to represent what you want to store. So it's really just these 2 types of elements in ZAR. The arrays themselves, these are actually not one file. They're what makes up an array is many files. So when you point to an array, really you're pointing to a directory that contains an array, and that has a couple of different types of files in it. There's a metadata file that's always named .zarray, and this contains a JSON data that specifies the details of the chunks and how they're stored. So this is multi-dimensional data. It's quite similar to NumPy arrays, if you've worked with those before. We have a shape here. So this is a critical, the size of each of the dimensions. In this case, we have 5 dimensions. Some of these are just size of 1. And then we have a couple of other larger, in this case, X and Y. So 5-dimensional shape. And then the other bit of data that's interesting is the size of the chunks. So this is how the data is split up. So similar to like how you'd split up a big image into tiles. These are but these chunks themselves can be multidimensional. And so you have the size of the chunks here. And then there's some other data here, such as the type of data and other details. So you don't have to write this data yourself. The various ZAR libraries will write this, but you can, it's human readable at least. So that specifies the kind of chunks that you're storing. And then in the same directory, what you'll find is the chunks themselves. And in this case, if it's a 5-dimensional array, the chunks will be named in this sort of format, specifying the position of that chunk within the indexes within the larger array. So you can see these. So you'll typically have a large number of these chunks alongside this, and so those files together make up the ZAR array, and that's a directory. So to specify some more complex kinds of data, we can organize these directories into other directories called groups, and groups have a file in called zatras, and this is just a JSON file that you can pretty much put in what you want to describe the type of data that you're storing. So really our work in coming up with the current OMI ZAR spec is how to organize these arrays into groups and the type of metadata that we store in them to represent the data we want to represent. Okay, so we've got images, labels, and high content screening. I'll just cover images and labels now, and the high content screening data we'll come back to later if anyone's interested. So images, again, an OMI ZAR image is really a path to a directory. And although often you'll see this named as something .zar, really the name of the directory doesn't tell you anything. It's really what's in the directory that tells you it's a ZAR file. So, and in the top level directory, you'll get this zatras file. And this again, this is JSON data. And what's important here is this object called multi-scales. And this tells you how to find the various resolutions of a pyramid. So in fact, even for files that are not large enough to really justify a multi-resolutions of pyramids of data, we still always have this multi-scales object here. And it may have one or more data sets, different downsamplings, different pyramids within it. In this one, we have, and then underneath the data sets, we have a path, which is how to find, it's basically the directory that stores the array itself. So in this case, we've got 3 paths, and these will typically be the largest resolution of the pyramid, and then subsequent downsamplings of that pyramid. So that tells you where the data is. There's one other bit of information in here that's typically a Mero. So we're exporting this data often from a Mero. We have the data in this kind of format already. So this is kind of a shortcut that we've taken to use an existing kind of layout of the data to store what's useful for rendering this image. And this is one or more channels to view of the image, and we'll have rendering settings and a color and maybe a name of each of the channels. Okay, so that's roughly what an image metadata looks like. And then below that, so if you've got the image.zar directory, and then a path of the highest resolution, in this case 0, then underneath that directory, you'll find the array of ZAR data, the Z array that says the size of it. So notice that the size of the image is not stored in the image metadata at the top here. This is actually within the ZAR, the ZAR specification data. It tells you the size, the shape of this array and size of the chunks. So that's really the essence of an OMI-ZAR image. So I'm going to just run through a little bit of code here just to show an example of loading some sample data. So this is Python, and we're using here, someone already mentioned this, Dask arrays to load the data. So anyone who's done much in Python will be familiar with NumPy arrays, and a Dask array is kind of like this idea of a virtual stack. It's kind of a virtual NumPy array, and you can specify the size of the array. You can pass these arrays around. You can crop them and stack them, all without actually loading the data themselves. Only when you actually need to view or to analyze the data, you can choose to actually load the data, and that will require get the data off disk. So again, convention of import NumPy as MP, import Dask as DA. So that's a Dask array. Okay, but let's just start with this sample data. So I'll show you in a minute where there's a whole bunch of example files to work with if you want to try working with these different ZAR data. We've generated a bit of sample data. So let's just make sure this is imported. So we're going to take that data and go through all the resolutions, get a Dask array for each resolution. So this is a Dask array from ZAR. And so we have in our hands a list of five-dimensional arrays that represent the levels of this pyramid. And then if we display them, we can just display them in the notebook. Gives you a nice representation of what this looks like. So you can get a good feel for the size of the data. So this is a quite large single plane EM image. And so the size of the array itself is 26 gigabytes. It's 93,000 by 144,000 pixels. The chunks are 1K by 1K. And so you have a good number of chunks. This is the highest resolution. And then downsampling by a factor of 2 each time, we've got all the way down to, I think, 10 levels of resolution in this case. And then the smallest one fits into a single tile, let's say 141 by 91 pixels. Okay, so we've got a stack of kind of virtual stack of data in our hands. Let's try viewing it. I'm just going to pick the last in the list of arrays there. And this is a 5-dimensional array to show just a single plane. We're going to slice, just take the first index, in fact, the only index of that array to get a two-dimensional array. And then, so actually I can, these have already been run. This is showing the output here. So this is a low resolution thumbnail. You can see it's very pixelated, 91 by 141 pixels. The next example here is basically just picking a couple more levels. This is third from the top. And again, just showing the entire, still at this level, we've got the entire array on one tile. Now what we're going to do with this code is basically iterate through the whole pyramid, starting at the smallest resolution. So we can't, as we zoom in, we can't show the entire plane. So what we're doing here, so here's our region that we want to crop here, 500 by 300 pixels. We can basically work out the coordinates that we want to crop in X and Y. And then we can crop this large 2D array into a much smaller region. At this stage, we still haven't loaded the pixel data. But then when we come to show the image itself, that's when we're loading the pixel data. So again, I can scroll through the output from that code and see start off at low resolution and then higher resolution, and the resolution is shown each time is 6, 5, 4. So we're getting higher resolution data as we zoom in. Down all the way. So all the way down to the 0 resolution. So this is the original resolution, highest resolution of that data. Okay, so that's a single channel, large X and Y, but a single channel, single Z and T image. But you can work with all different types of data in the same way. Okay, any questions at this point before I go on to talk about labels?
Sorry, with the data files, when you generate them, I'm assuming it's up to you guys to choose how you, what pyramid levels you're essentially storing. Is that correct?
Yeah, so the user gets to decide how many pyramid levels, the scale between the pyramid levels, the chunks of each pyramid level, and the compression. So all of that is basically up to whoever's writing the data.
Got it. Okay, thanks.
I have a question, like, can we, should we preserve like dimensionality of the data inside the ZAR array, or we can also include some projections which we are most interested in?
Yeah, so I think that would end up being separate images. You know, so if I'm understanding correctly, if you figure your data in one dimension, sorry, one might have. Does that fix it? Yeah. So you know, you would have the original 4 or 5D data, and then you would have another image, which is the projection, and then eventually, what there'll be metadata that you put on one or both of the images to say these two things are related, right? But it's a separate quantity at the moment.
Okay. Yep, muted. Okay. So that's covered images. Labels are, you might know them as masks, or certainly in Amero we call these masks. These are basically regions of interest that are not polygons, but these are not vector. These are pixel-based regions of interest. Typically, what you'd get from an image segmentation. And so it's quite convenient having, since ZAR is designed for working with images, and we know how to work with images. Labels are really just another type of image. They have a pixel value of 0 for all the background regions, and an integer pixel value for each mask, and all the pixel values are the same to represent a single object. So for the labels for a particular image will be stored under the image directory. So you've got an image.zar directory, and you've got labels you want to store on that image. These are under labels directory, and the metadata within that labels directory, this ZAttrs, will give you one or more paths to the labels. So you may want to store more than one labels object on an image. So we can then go use this to follow the path. Again, 0 would be the simplest possible name. You might have other names here, but this specifies the directory under the labels path, which we can find the chunked data. So again, we're starting here with a path to an image. We can look under that image to get the labels data. Again, we're looking for this multi-scales object, and we can go through that to find the path to the actual the ZAR array. So we're going to take the first ZAR array in that list, and then basically load that again into a Dask array. So we want to do a bit more work here to display it. So the pixel values within this array are So if we just render it straight out, we won't be able to see the different pixel values between these different labels because they are quite large values, but very close to each other. And I'll show you that in a second. So what we want to do is we want to find the range of pixel values so we can use this for rendering. And as I said, this is a virtual ND array, a 2D array in this case. In order that we can actually read the max and min pixel data on it, we have to do this dask array compute, which will actually turn the virtual stack into an actual stack of data, so we can then get the values out of it. So that's one thing we're doing differently here. Let me just show you what that looks like. I'm just going to play this. And that's going to load this data. And you can see there's a bunch of masks on an image. Each one has its own particular gray level. Each one has a different pixel value. And here I printed out the minimum and maximum values. So these are, this is actually data coming from an instance of Amero. And in this case, I've chosen to export it using the ID of the mask within Amero as a unique pixel value. And that's useful if I want to stitch these images together into a high content screen, as I have examples of showing that. But you could equally well just start off going from one and increment each higher. As long as each label has a unique pixel value, doesn't really matter what that is. Okay, so that's a mask, and again, the dimensionality of a mask will typically be the same as the dimensionality of the image that it represents. Actually, one nice thing about this, if you've got a very large image with sparse data, the specification of ZAR allows you to leave, if you've got a whole tile that has effectively got nothing on it and is all zeros, you can actually just have that tile completely missing from the directory of chunks, and that will be understood to be basically a tile of zeros. So you don't actually have to store that empty tile on disk. Okay, so that's how we store labels. Just a little bit of extra metadata associated with labels. So if you look in this path to a labels directory, the Z attrs metadata within there, we've already used the multiscales metadata for actually finding the data itself. But we also have an image label object in here. And this stores a couple of interesting properties. We've got colors. We want to store colors for each label. This is a list of objects. Each object has this label value that specifies the pixel value that this applies to. So each one applies to a different label. And then here we've got the color for that label. And the properties works in a very similar way, except in this case, so we've got the label value to say which label we're talking about with this data. And then we've got kind of arbitrary pairs here of key and value. So we can store additional customized data, text and other values for that label. Okay, so I think that sums up gives you at least an overview of the specification for storing images and masks. There's some more here about high content screening. I'm not sure there's many people interested in that, so we can come back to that later this time, and depending on interest. So I'm just going to go back to the document that I posted earlier, and just follow a couple of the links in here. So really, what we've been trying to do to test out how valid this spec is. So we don't have our own data or our own analysis goals in order to drive this, but we want to be able to write some tools use some tools to visualize this data to give us an idea that we're kind of on the right track and that we can work with this data. So we've done this in a couple of different ways. Actually, this whole first section here, viewing this data using JavaScript in the browser, is really the work of Trevor Mance at Harvard Med School. And so there's a couple of different libraries here that he's developed in his lab, or not his lab, the Gerenberg lab, along with Ilan Gold. And so the simplest case here is a very lightweight library called ZAR Lite. And so we can just, if we want to do some simple loading of data, and we just want to display it in a web page, we can use this library. It's loading. Yeah. Okay. So again, this is basically doing similar in JavaScript as we've just seen doing in Python. We're loading a pyramid by getting JSON data for this path. We're getting the model scales, loading a pyramid of ZAR levels, passing it, in this case, we can work with the lowest level of the pyramid because it's not a very big image. And this draw method here basically gets the raw data for the dimension that we're interested in. Here are the 5 dimensions, and it's just rendering that. It's just reading the pixel data out of that, and then just applying that to a canvas, so we can see the RGB values on that canvas. This is saturated because we're actually reading 16-bit data and applying it to an 8-bit image. So in the next example, we just go one step further than that and use some of that rendering data that I mentioned, the Amero rendering settings that we have available to help sort of rendering that image. Okay, so here where we're loading it, we get the pyramid, we get these JSON data. We're getting the channels, the Amero channels, going through the channels, basically getting the min and max values, and passing it to this view method so that I won't go into too much detail. But here where we're basically painting this data into an 8-bit, we're scaling it according to those min and max values. But this works. It's kind of the right intensity for us to view, is not saturated. But this is really not very performant, and if we wanted to do this for larger data and adjust it as we're playing with the rendering settings, this really wouldn't scale very well. But it's a useful light example if you just want to get the pixel data in hand. So a more production-ready viewer is this Visar viewer, and I'll just show this. The best way to show this is to go to this list of example images that I mentioned before. Let's open this in a new tab. So this is a bunch of sample images that we've chosen from the image data resource, the IDR. We're using the Visar viewer to show these here. I'll just scroll through the list a little bit. So we've got a bunch of different images. The columns here, the X, Y, Z, CT dimensions. So we've got a mixture of very large single plane XY images, some spinning disk data, large X, Y, Z, T, so large in every dimension apart from channels. So this is coming from the Keller lab. And yeah, just a bunch of, so this is the image that I showed before. So let's just take, okay, I'll first just give show this image that's already at the top here. So we've got a much better experience here. We can zoom in, adjust the rendering settings on the fly, and so the rendering is happening in quite a performant manner on the client here. It's using the WebGL technology. I've just gone through the Z sections, and again, it's loading the different levels of the pyramid as needed. Let's just go back to this image that we've seen before. So I'm going to open this in. I can just click here and it'll open it in the viewer above. But if you want to view a full size, you can open this in a. Just open that link in the new tab. Okay, so again, we're starting at the lowest resolution and zooming in. And what's interesting here, might be useful to see what's going on. If you right click and inspect, or might be inspect element in different browsers. But the nice thing about the web here is we can go to the network tab, and we can actually see the various data that's being loaded. As we zoom around here, you can see these chunks of data. And if I hover over this, you can see the path to here. So it's got a /5 in it. So we're at the 5th resolution of the pyramid. As we go on in here, we're loading more chunks all the way down to, so at the 3rd level here, and then all the way to zoom in. Okay, so we're viewing a large amount of data. We're loading the raw data, doing and the rendering of that raw pixel data on the fly. Okay, so that's this bizarre viewer. Yep, and you can actually, let's just show that one more time. So you notice that the viewer itself here is just statically hosted at this address. So this is Harvard Med School's hosting this, and this is loading the JavaScript and the HTML, everything from that location. And then you can basically point this at any image that you want to. So if you put up your data into a location of your choosing, you can use their viewer hosted at this location, or you can host your own if you want to. But this is basically their viewer pointing at the data we want to load. So we can use. So you can see in the URL here, that's the starting URL, the same URL that I used in the Python example before. And again, if you just want to copy that URL, that's what this link on the right-hand side is. If you want to copy that URL to use in some other tool.
Will, I'm assuming that's all client side, that their servers are not doing anything extra.
So this is completely statically hosted from their side, and again, and the pixel data itself is coming straight off this S3. There's no server there either. So yeah, there's no server involved in any of this,
cool.
Which is why it's designed to scale well. So okay, so that's the Visar viewer. That's our current list of sample data. I should say these are all this blog post, and kind of the reason it's a blog post, this is version 1, kind of the current state of the spec. And all these URLs will stay up in that location. You'll notice the URL to that data has version 0.1 in it. So that's the version of the Visar spec. And that will be a constant URL. If we generate more data for another spec, that'll be at a different location. Okay. So that's Visar viewer, and then briefly I'll show Nepari. And again, let's just use this. I can copy this URL again and use this in Nepari. Oops. So, and again, you can do that with any of those images. So this is using so instead of running Nepari itself to read this data, this is all encoded within this Nepari plugin, OME-zar. So you can pip install OME_zar, as described at the top of the page. And then that will be picked up and used to open any file that the OME-zar plugin understands as a zar file. So you can then pass Nepari a URL like this, and again, it doesn't need to be called .zar. What's important is that this file contains a Z-attributes JSON data that has multi-scales object in it. That's kind of the definition of the file that OME-zar will recognize. So let's go back to running this notebook. I can just stop this notebook and run Nepari. Okay. So this is again, it's doing the same things we did in the Python example before. It's creating a pyramid, a list of Dask arrays. In passing those list of Dask arrays to Nepari, we're not loading any data up front, but Nepari is then able to request the pixel data that it needs from that kind of virtual stack, and those tiles will be loaded as needed. Okay, so again, same image you've seen before, and the very similar behavior. Again, this is at this time we're in Nepari instead of Visar. Okay. Right, so that's most of what I want to show. So I'll just go show one more example of labels data in Nepari. So we haven't yet supported labels in the Visar viewer. That's still to come. So I'm going to choose this example at the end of here, which is the labels, an image with labels. It's the same labels that we loaded before in the Python notebook. So actually, this is an image within a plate. I could show that later, but this is basically the plate, and then in the A1 well first field of a plate. So again, you can see that this is an image we're loading, but the image, the path to this image doesn't end in zar. It's just but that still contains the image data. Okay. So you don't see the Nepari loading icon as in my other window. Okay, so here we've got the two levels of pixel data, and the plugin is returning this third layer here, which is the labels for this image. And if I mouse over individual labels, you can see in the bottom of the viewer the extra properties. So these are values stored in the custom properties metadata for each label. And so they've got a bunch of things. They've got some IDs in them because they've come from Amero, and we've used those IDs to add additional data to them. And they've got some other interesting data, like some area. So maybe just briefly, again, this is not a Nefari tutorial, but I'll just paste in some of this code here, which makes use of that properties for each of these labels to show some of those labels in different colors. So I'm importing NumPy. I'm choosing the layer that's the last layer in this stack. That's the top layer. I can, if I want to view what those layers, or what the properties look like on those layers, I can see we've got various values for IDs, min. So these are kind of in pixel intensities for the data within these arrays. And I'm going to use this area, the area of these different labels in order to view them differently. Let's go back to here, just copy this and explain what that's doing. Okay, so we're basically going to go through, let's make this a little bit bigger. Going to go through all the list of properties, and depending on the value of each property, we're going to set the color on the corresponding label. And so above a certain threshold, it's going to be red, below it's going to be green, and then otherwise it'll be white. Okay, so that's just updated. You know, I can now see labels that have particular properties on them. Okay, I'm gonna finish there, and yeah, any questions on any of that before I hand back?
I have a question about, I guess, how other types of metadata fit into this for images. So for example, if you want to say what the scaling factor is for the different scales and multi-scale, and then maybe give pixels or voxels real units like microns or something.
Yeah, so for anyone who followed along with the original specification of the multi-resolution, so that was all proposed, and then there were too many dissenting votes. So I kind of kicked it out of the spec and said, let's deal with that later. Yeah, it keeps getting pushed down the list. So Tischer, in what he'll be showing next, has basically made a proposal. So they've pulled in the scale and voxel information from In5 and put it in a ZAR. So if anyone wants to take a look at that and kind of say, you know, can we start to agree on a proposal? That would be great, and then get that through the process. I mean, it's the same issue for us. We would love to have it in all of the data. Yeah, it needs someone to help champion it and just get it through the process. I imagine that'll happen in the new year, you know, so maybe we try to get it done in Q1, you know, something like that. I know Juan is really pushing for it, Juan and Draga, I assume Solfeld, you know, Kevin, if you have a use case, it just takes a small handful of people who are all looking at something and going, yeah, we all agree, and then we're good.
Okay, cool. Yeah, because it just seems for interoperability between different viewers in terms of being able to, yeah, display things properly, especially with mixed data sets. That seems pretty important.
Yeah, which is probably why there are so many opinions, you know, so that's agreed.
I was just curious, and there are two aspects to this. One was, I'm assuming labels are things like segmentations and that kind of stuff. So that's what that layer is for, correct? And then the other one is, and this is, if this is out of scope, just say so. With RGB or RGBA or whatever type images, the dimensions and how you store that, is there a consensus? Like I've noticed in TIFF file, it's done with like channels, and samples are independent. So RGB would be samples, whereas, you know, sometimes they're, I mean, it's a bit of a little, I find it irritating, let's just say. And I'm curious if there is a general accepted way to handle that.
Yeah, so I'm open to thoughts as well. So I discussed this with Melissa recently about how OME TIFF does it. And basically the TIFF model of samples per channel is basically a burden that we've been carrying around for many years. So if we could not make some of the same mistakes going into the NGFF, I'd be really happy. If someone knows a use case where it's definitely needed, it may be that eventually it arises as a need. At the moment, I think we would try to keep all of that out.
You're hoping to not have a samples channel, for example.
Yeah.
Okay. Because we don't use them internally, but we find it keeps popping up with external users who have these RGBA or RGB files with channels. Like I got a support request for one with 7 channels, which was RGB, and how you handle that, you either explode it out in the channels to 21 or you handle it as samples, and it's the right way kind of thing was definitely intriguing.
I mean, for me, this goes back to the dimensionality question we had earlier. So if things can be modeled as channels, then just use the channel dimension naturally, so for whatever matches your data. If there's a new dimension that needs defining, let's do that. But, model it as a full top level concept and then make use of it.
Okay, thanks.
I just want to say I posted something in the Slack, so we had actually preparing this workshop a GitHub repo, which is essentially only the discussion. So if you guys I think it can be fun to read it. I mean, there you see kind of also what's already fixed and what one can still discuss and so on. Yeah. So I mean, there's many issues, and then
this is the one you put in our tutor room. I assume you want me to put it in the main room.
Yes.
Okay.
Sorry,
I'll do that.
guys. Okay. Yeah. In a second.
There it is.
Yeah. I think it can be, if you really want to get into this, it can be fun maybe.
Yeah. So this is the scale information, Kevin. So and opinions from everyone welcome. Anyone else from the Python and JavaScript side? So otherwise, we're slowly moving away from those. We can always come back later. I mean, the links, so especially the JavaScript, it's very hackable. So those JS fiddle links, if you want to start just playing with JavaScript, that's a good place to start, just because you can change a variable and the image updates. So even if you're a complete newbie. Otherwise, I propose kind of a 10-minute break. So give us till 20 to the hour. And then we'll do some stretches, open your windows, get fresh air, wake back up. Yeah, do dancing if you want, whatever, anything goes. Go look for Hebidas, whatever it takes. All right, I'll stop the recording or pause it at least. See everyone in 10 minutes. Leave it up to you, Tishy, how long you want to wait to get started.
So hi, welcome back. If you're back, can you click on participants and then in the lower, where all these icons are, click on yes?
We could hold up your famous done or still working on signs from the workshops.
I mean, that's the equivalent, I think. So not everybody was doing it yet.
Like in a real workshop.
So that can mean many things. So maybe for everybody that is here already, so I think the next two things should be interactive. I mean, maybe the things were already interactive, but now even more. So you could already start Fiji. I hope you installed this update site that we had in the instructions, this Mobi beta. If yes, then start Fiji, because then you can really click along now, because that should be very simple. If not, then maybe just watch, yeah. Okay, I clear again, and maybe you can again click yes now if you have your Fiji on. Just opened on your screen somehow. At least that I can see how many people would like to follow. Okay, that's good. Perfect, wow, great. Okay, so what I will show now, and the ones of you that read maybe this issue that I wrote, I think is actually kind of fun to read it. So I was working the last 2 weeks on reading these SAR files into Fiji. And so I will show now how far I got. And I have to say, I took a library from Stefan Saalfeld as a base, so the N5 library, and then also Josh added something or merged 2 things there so that you can now use N5 to read SAR. So, I mean, if you care, you have heard it, but so I basically had not to do all the work. So what I will do now is, first of all, I should share my screen. Share. Okay, so I will copy and paste a bit of code into the Slack. Copy, paste. Okay. Do you all see the code in the Slack? Yes. Okay. So now what we should all do next, click on Fiji and say File, New Script. Okay. File, New Script. And then click on the script window, and then in the language menu say Groovy. So Groovy is essentially the scripting language of Java. I mean, you can use others, but I think that's kind of the most normal one. Okay. And then you copy and paste the code from the Slack into this window. Oops. And you can press run. Okay, and now all of you should see a big data viewer window with some sort of image, okay? Can you kind of make some gesture if it works for someone? Yeah, okay, good. Okay, then before we look at this image, let's first look at the code. So the first line is not, okay, did this console window pop up for everyone also? If not, then go click on ImageJ, window, console. So this is where you can see text output, like from standard out kind of stuff, like print statements and so on in Fiji. That's kind of useful. So the first line of code is actually telling the software to print into the console window when it's reading data from the object stores. So what you actually see here is how it's fetching these different chunks at which coordinates from these different paths in this object store. Okay, and the S0 means highest resolution, S1 is one downsampled. And here it also tells you how many voxels it was downloading from the internet in how many milliseconds. So, okay, I'm jumping a bit, sorry. Let's go back to the code. So the whole point is, and this is a bit similar to just pasting this whole web like link somewhere, here it's just split up. So we generate a reader. And the first thing is we have to tell it like what's the kind of server. I mean, it's not really a server, I think, but where is this kind of S3 thing hosted, right? So this is called the service endpoint in this kind of language. And this is actually a one that we host at EMBL. Then this thing is not important, I think. We ignore it. Okay, I think you can also put nothing there and it should work. I don't know, but let's ignore it. It's not important. What's important is the next thing. So this is in this object store language, so-called bucket. And you can put stuff into buckets. And the reason why this is important, this is the level on which you can manage access rights if you really want to. So you could actually, in my case, it's obviously now open to everyone, but you could distribute keys. So we sometimes do this if there's unpublished projects, but we still want to share it with our outside collaborators. We make such a bucket, but then you can have access keys. So that can be relevant, yeah. And then in this bucket, you can have multiple keys, and these keys are your data. So this is, for example, a ZAR file that contains some data about myosin expression in this Platinares animal that I was talking about. So we tell the reader to read this thing, and then we tell Big Data Viewer to show this thing, okay? Are there any questions maybe here for this code? Kind of self-explanatory almost, I think. So now I don't know how well you are with Big Data Viewer, but what's the kind of cool thing about this? If you click with the left mouse button, hold it pressed, and then move, you slice through different planes. So you can change the angle through which you slice through the data. So this is the thing you cannot do with the ImageJ viewer. You also up here you see a animation of this, and you see how it's fetching now the chunks that it needs for this from the object store, right? So this list is now moving, and it's not moving anymore, and the reason is that there's caching. Okay, so BDV internally or ImageLib2, I don't know, maybe some of you have done the practical, they are a lot about caching stuff, so you don't load it again if you have already loaded it, unless you are running out of RAM. Okay, if this is an 8 terabyte data set, then you cannot cache everything, but they have quite good logic to do this automatically for you. Mouse wheel would be scrolling up and down through this. Okay, so this is an example how you can just load a simple 3D volume from the internet into Big Data Viewer. So far, any questions? If not, then let's add 2 more lines of code to this. So I'll paste them in the Slack. And then let's copy them beneath what we have already. And this is now not like in Python, you can execute only these 2 lines. We have to run everything from the start. At the moment, I mean, there will, okay, so at the moment we have to do this. So let's also close Big Data Viewer because we don't want to have it open 2 times. And then I'll explain what we added. So we are reading now another file. And that's now, I called it EM and labels, okay? And that's now a bit of the special thing. I think that's for me and you. So far the specification actually decides you put your segmentation into the same file as the raw data, okay? For me, this is a new concept. I always have my label mask image extra, right? This is one OME TIFF with the label mask and another OME TIFF with my raw data, but somehow the current idea is let's keep things together that belong together. So let's put the labels inside, right? So maybe we can discuss pros and cons. But anyway, that's how it is at the moment. So this thing is actually two things. But Big Data Viewer is clever enough to show both. The point is now actually, and this was what I said at the beginning, we want to show this on top of this other data. So this is the expression of myosin in this EM data set, and they have completely different scales. So this is at the resolution of maybe 10 nanometer, and this is just 500. But the good thing about BDV is it can natively do this. It's really a viewer that's based on a physical coordinate system and not on a voxel grid coordinate system. So very different concept than the ImageJ1 viewer. So BDV is really good. It just wants to know where a pixel belongs to in physical space. And then it can show all kinds of things on top of each other. That's why it's really great for this kind of things. And you don't have to doubly like upscale or downscale data. And this lengthy expression here just tells it, okay, please don't open a new Big Data Viewer window, but just add it on top of the one where you already were showing the myosin. Okay, so this is this add to, and from this myosin Big Data Viewer source, you get the handle in which Big Data Viewer window this is shown currently. So if we run this, it's reading the stuff from the internet. Okay, so now we have actually 3 images. And you can here, if you go with your mouse all the way to the right, this is actually a new thing in Big Data Viewer. Some of you might not know it yet. Considerable improvement of the user interface. Feeling the pressure of Napari. So here you can toggle on and off different stuff. So for example, we could only look at the EM raw data. And you can even change the color of stuff and so on, okay? Okay, I'll zoom in a little bit. And you see how it looks still not so nice. And then if we wait a little bit, the resolution will improve like we've seen before. Okay, there's one particularity I want to talk about quickly. So if we're now not only loading data like this, but like this, the voxels that we render on the screen, they don't fall exactly on voxels in the raw data, right? They can be kind of exactly between 2 voxels. So if you then want to have the best kind of visualization experience, actually, if you render this guy, you want to have a proper interpolation of all the voxels that kind of next to it. So in Big Data Viewer, you can do this if you press the I key. So I stands for interpolate. And then it's becoming smoother because it's doing all this math for you. So to deal with the fact that we're not on the voxel grid, right? And then it looks even a bit more beautiful. Technicality, but I think I mentioned it because you may hit this if you really start working with this. Let's look at now this label mask image. So there are now 2 problems with it. First of all, the coloring is quite boring, right? You don't want to color your things black and white if it's a label mask because the intensities are actually meaningless. The other problem is if you interpolate a label mask image, then where 2 objects are close to each other, like object number 1000 and object number 500, there's all of a sudden an object number 800 created at the boundary, which is basically not what you want. So you have to then, and this is not possible in Big Data Viewer, you have to tell it, don't do this for the label masks, okay? Don't interpolate because you will create nonsense. So maybe first, or no, I will first, okay. And to deal with this, because this is not natively in Big Data Viewer at the moment, we have to add another line of code. That's some code that I wrote. So let's do this, and then I come back to questions. So let's close Big Data Viewer again, and I'll add the final line of code to the Slack. And then I copy it from the Slack into the window. So what this line of code is doing, so these are two images. So image 0 and image 1. Image 0 is the EM and image 1 is the label mask. So it's grabbing the second one, the label source, and does some stuff to it that it's properly shown as a label mask. Okay, so if I run this now, so first thing it's doing, it's applying a random color lookup table so that all the cells that are actually were beautifully segmented by Konstantin Pape. His name was already mentioned a few times. He's a really good guy, as you can see. He can do many things. Yeah, so this is a much more appropriate lookup table for the label mask, which is nothing in BDV at the moment, but hopefully we can add it soon. Otherwise, you have to use my code. Okay, final thing is, typical problem you have here is that you have color clashes. So for example, here are two cells where I, with my additional color blindness, have a hard time saying, are they now really different or not? And this is always a challenge that you have with the label masks. So what I added is if you press Control and L, so L for lookup table, it's actually trying different, it's shuffling the colors. So for me now something is a bit hanging, but shouldn't be necessarily for you. So does it work? If you press Control L, it will change the coloring. Okay, I don't know. For me, it's now a bit stuck. I have to say this is something that sometimes happens. So I think if you just download all this stuff from the cloud, sometimes things still get a bit stuck. And then I just have to restart it. But then normally the second time I do it, it works. So it's busy doing.
So
yeah,
when you,
actually now,
quick question, when you change the lookup table, it seems to go from low res to high res again very quickly. Is that just a big data viewer thing where it's always, no matter what, loading low res first and then realizing it's already got the high res and puts that on top?
It's a good question. I think I also sometimes honestly have the feeling it's doing a bit too much. If you update something, it's reloading all kind of stuff. So maybe, yes, yeah. I could be room for improvement, actually, I think, in that sense.
I mean,
but
this kind of holds for anything when you're loading data from the cloud that, you know, you start to feel the pain of every small little inefficiency. So as users, I would just encourage everyone, you know, give that feedback to whoever's developing the tool, whether it's Napari or BDV or, you know, whatever. Sometimes they then have to go back to a lower level library like Dask. You know, it's going to be a process until we're already across the board to just load data from S3. It's yeah, it's challenging.
Yeah, I guess my biggest, the reason I was asking that was I wasn't sure if it was reloading data from S3 when you change the lookup table, or if it was just using what was cached already. It seems like it's using what's cached and not reloading it or
it should, yeah. It should. So I was a bit surprised that when I changed the color that I had the feeling there was some activity down here. So maybe having this lock on is not so bad, yeah. So maybe there's still something not going properly because it shouldn't reload stuff from S3 when I change the color. So, but that, I mean, now it's doing it smoothly, yeah. Oh, but now it's going back to, so I don't know. I think, yes, now it's okay again. Anyway, yeah, but this is our, yeah, as just said, we are all still, you know. but I think it's already pretty cool, to be honest. So I mean, like when at EMBL, it's really, people love this because you can just share like small scripts and everybody in the world can look at this data. Yeah. So I mean, I'm quite excited, to be honest. I mean, even cooler, of course, with this Visar, when you can just do this in the internet or in Apari or whatever. So I think the point we want to make also is with this workshop, like it works in all, we think, 3 major platforms, right? And it's always the same data model. So, which I think is quite exciting. So, okay, any other questions for me for the ImageJ part? If not, then we come now to a practical session where you can actually really create your own ZAR file and upload it to the internet and look at it, which I think is a really good practical. So I hope we can all follow along, and Josh goes slow enough.
Yeah, when you say it that way, it almost sounds interesting. So this is the behind the scenes part of everything, right? So the previous session, I sold it as being very boring, and so Tischer did the sales pitch for me. So let's see, where do we want to start? We want, first of all, to get you, I'll just share my whole screen and then you can see the links that I'm sending you. I guess first of all, you'll see Tischer. He disappeared. Okay, so I mean, this is just the repository that I had open, or had in the installation instructions. So do go ahead and, did someone already paste it? No. Okay. Go ahead and clone that. And actually, let me do the yes trick from Tischer. Say yes, please, if you, in the participants list, if you have my Conda environment activated.
Yes.
Verbal works too. Cool. Okay, I'm going to assume it's working for almost everyone. If you're having problems, please ping in Slack and someone will help you. Maybe not me. Okay, so I need to get caught up. I've already activated, so I'll do all those steps so everyone can see. Activate Josh. Sounds a little bit weird. And then I'm going to start with the cloud notebook. So what this really comes down to is getting our heads around what is S3, right? I'm going to open up the notebook, which if we take a step back, if you remember, I asked, what is the cloud? Because that's not clear. So when I say the cloud, or when we say the cloud, what we're really talking about is object storage. So Dropbox and Google Drive are sometimes referred to as cloud, cloud storage, but they're very different. You basically, the only thing you can do is download all the data. So let's not talk about those. The only providers we're really talking about are, in the commercial sense, Amazon, Google, Microsoft Azure, right? So these are companies that provide object storage. There are scientific providers. So we get ours from EBI. You may have S3 from your institute, for example. Usually that will be some other less well-known commercial provider, maybe a Dell or a IBM. I mean, they're well-known, but they're not well-known as cloud providers. There are open source projects where you can run your own S3 storage. So we'll be actually working with, the client from MinIO, if this finally loads. And it is actually as simple as, so I created this data directory earlier. Let me touch a hello file. And if I run minio server and the current directory, then that actually starts up an S3 server. So,
Josh, can you be more explicit when you want people to also do it and when they just should listen.
Yeah,
Just
don't follow along yet because you haven't installed that. I'm still explaining. So you don't have to do anything until we get to the notebook. For some reason, the data is not in there. I don't know what that means, but you can run your own server, and it may be something that we're actually doing from our side with Omero. So you need a provider, and you need someone who provides you high performance S3. Now for testing, everything we're going to do, we're going to be using kind of slow scratch S3, which works for testing. But eventually you need someone to give you object storage, and it needs to be good, right? So those are the highest level caveats that I can give you. And the reason that we're doing all this is because more and more institutes are saying that storage must be an object storage. So your data must be an object storage. That's the only storage they're going to give you. So that's just something to be aware of, is that that's happening more and more institute-wide. That's where the investments are going. And we're basically getting ready for the day when you must put your data into the cloud. Okay, let's go through the notebook. You can just follow along top to bottom to begin with, and then we'll start. I'll ask you to edit some of the cells and run it with a different value. Okay. So I've gone through the top bit, which is what is object storage. We're only looking at S3 for the purposes of the rest of this demonstration, and we're only using one client, MC, from the MinIO project. There are others, they have trade-offs, right? So one of the reasons to use MC is because it comes pre-configured with an endpoint that all of you can write to called play. So if you list the configuration, so I ran this cell, you can see that there's play there. This is the URL. So we've been talking about two endpoints so far. We've seen Tischies, which is s3emble.de, and the IDR S3, which is s3.embassy.ebi.ac.uk. Those are production S3s that you can't write to, but play you can write to. So this is your connection information. We all have the same one. So there's an access key and a secret key that basically lets you do whatever you want with play MinIO. So while I'm talking, if you want to go around and just figure out how to do stuff, I don't think you can destroy it too badly, but if you do, let us know. That's kind of interesting. Tischi covered the concept of buckets. So we need to create a bucket. It should have already exist. I checked about an hour ago, but someone could delete it. So this is always kind of dangerous when you're doing a demo, but our bucket is I2K 2020. And then I'm going to set it to be public. Okay, so that means any of the code that we've looked at so far today, you could run against our bucket. You could look at JavaScript against the bucket, Napari against the bucket, BDB against the bucket. All the clients would work. And let's see what's currently in it. So you can see all the hellos. I'm going to ask you to create a hello by the time we're done with all of this. So if you want to go ahead and figure out how to write the hello, feel free. Oh, and someone even uploaded their own GIF. Good job. So one of the, well, I guess I'll get to the caveat in a second. So let's go ahead and do your first upload. You use something called copy. Makes sense. MC actually can replace a lot of what you do even locally. So if you want to get in the habit of using it for your own data locally, and then you can also use it remotely to various servers. So I think in my configuration, I have something like 7 or 8 different S3 storage locations. So endpoints that I use. And MC can let you copy between the different S3s, upload from local to S3 or download from S3 locally, right? So it becomes quite a natural tool for moving data around. Right, so if I do hello, so if it doesn't recognize, so here's my server. So if I had started this with play, I can move on S3 hello to goodbye. And then if we list the bucket again, we should see goodbye. And there's goodbye, right? If you want to download something, so I don't have currently a goodbye file locally, I'm pretty sure. I haven't done this before. Yeah, no directory. And then I can download goodbye into my current directory. And now I have it. So all that should be fairly straightforward. The cells should work for you as they exist. Is anyone having difficulties? I assume you can ping me, right? Okay.
Quick question.
Yeah.
MC just an alternative to, say, the standard AWS CLI interface, or is it doing more than that?
It's not doing a lot more. So for everyone's benefit, the two primary alternatives I know if you want to work with S3, as I'm doing here, so kind of from the command line to get your data somewhere, are rclone, which lots of people have good success with, and then the official AWS command line, which you can pip install it, pip install AWS CLI. The big downside of AWS, Jamie, is that they actively don't let you configure other endpoints.
Okay.
Which means you must always type -- endpoint URL HTTPS S3, the whole thing.
Got it. Okay.
Which is really annoying. And I'm almost positive they do that very consciously because they want people to use AWS servers, right?
Right. Okay. Thank you.
Yeah, no worries. So MC is just very, very short. Okay. So there are a couple of concepts that are different when it comes to S3. There are probably lots more that I don't know about either, but I want to get a couple of cross. One is the metadata. When you upload something to S3, it is actually its own individual object, right? It's object storage. You're creating this thing. And so you can do things like set attributes on the individual files. So I can say on my data, this is a plain file, or if it's JSON, I can say, represent this as JSON. I can tell you, we can tell the server how long they should cache the data. So there's a whole language of metadata that you can start adding to your S3. You can have objects automatically deleted after a period of time, et cetera, right? So basically everything you know about how POSIX works, so about how the file system works, you're putting data in a directory and you're giving a user access to it. There are new ways of doing all of that, and it's just something you need to be aware of, right? It's unfortunate. As I said in the last session, if I could have any wish, it would be that, well, when it comes to this stuff at least, it would be that S3 had a specification like POSIX does to make all of this stuff straightforward. Your mileage may vary, unfortunately. The other thing is the naming. So, everyone's off creating objects. Actually, let's see what objects we have now if everyone has, if we've gotten any more. Has anyone done anything really interesting? So there are couples, not everyone's uploaded a file. Tischer was here, so you should really have a go, put a file up there. And the one I'm really interested in is what happens when people start using subdirectories. So I'm going to go ahead and create a subdirectory and do Josh was here down deep. That works fine. Obviously uploading simple enough. This is the one case where the command line, the tools that you're using lie to you, right? So if I ask what's in I2K 2020, I see that there's a Josh directory and I don't see what's in it. If I pass the recursive flag, it shows me everything. It shows me all the images, lots of, where's the Josh directory? So here, Joshua's here. So the next caveat, and I think the last caveat, and then we'll kind of get off the basics of S3 and we'll actually look at generating data, is that this is something of a lie, right? Object storage fundamentally doesn't have directories. People want directories. We're used to directories. They show us directories, but there aren't really directories. And that's just something we all have to internalize, right? Each of your objects lives completely on its own. And so part of what we're doing with this NGFF specification is encoding in the metadata of the ZAR logic to find all the objects you need. You know, so if we look at the example that's here, this top level attributes knows about the multi-resolution. The multi-resolution knows how to find all of its chunks. That's all well defined because there are S3 servers which won't let you list the entire bucket for performance reasons. So basically everything that object storage does it for performance and cost. You know, and if so someone wants object storage that's as fast and as cheap as possible, you're going to lose functionality. And it's functionality that you would have access to in the file system, but you don't with object storage, right? So if you don't kind of get used to this concept of it's something different, it looks like a file system, but it's not a file system, you may make assumptions, or, you know, if you're a developer, develop code that's going to get slower and slower over time, right? So that's the message. That's one of the messages. Okay. Does anyone have any questions?
Maybe give a little example from when Josh and I were trying to prepare for the workshop. So we had a folder structure. There was one top layer, and this thing had just 3 subfolders, okay? But then beneath that, there were millions of files, all these small chunks. And I just did a list directory here and just wanted to see these 3 files, okay, that are just directly beneath it. But this command never came back to me, probably because it was going through all these millions of little keys, right? And that would not happen on the normal file system. If there's only 3 things, it comes back to you immediately, even if there's millions of other things. And then we could actually not figure out whether these 3 things are there or not. Yeah. So I mean, these kind of things, they behave slightly differently than on a file system, and that might be a practical problem. Yeah.
And I can give, so that's the one example we've run into. And actually during the previous session, here's another example. So for the purposes of the IDR, so the IDR public data exists here. I can give you, can I type this in? MC, I'll paste this for you to type it in if you want it yourself. MC config add host add EBI demo S3 embassy EBI ABC. Okay, it's going to ask me for a password and I ignore that. Okay, so this is going into Slack. And then you can type, then all of you can look at the IDR data. And it's all listed there. So I'll give you that. So that's fairly straightforward. I think that would actually scale quite well. But it's only our public data. Thank you. So the reason I have to do this from the console is because I'm not going to show you the next configuration because that has my password in it. So this is the IDR is the public data. And then for our submitters, we have an upload area where we give them out individualized passwords. So that's MC/ls IDR upload. IDR upload. So I'll just show you my area. IDR. And so my point actually is here that it's getting slower. And the reason that it's getting slower is that we run Minio here to give the users passwords, right? So Minio is basically a proxy. It lets you set up user passwords and start to act like your S3 is a file server. You know, it's FTP. You can let people upload things. The cost, though, is that you must then configure, you know, a check per directory. Like that was checking, does this person who's accessing have permission to list this directory? And then immediately the speed goes away, right? And it's kind of scary how quickly that happens. That doesn't return.
Yeah,
I think maybe that's just Heidelberg.
Yeah, I have slow internet on top of this.
Okay. It is a good deal of data. Okay. Yeah, so I think we've talked bad enough about S3 now. Any questions or thoughts before we move on to generating data?
So one question on just with this kind of thing, and I'm not sure if this is what you just said, but like something I remember doing when I was doing a lot of work with S3 was, and I'm trying to remember the exact details, but I'm pretty sure I mounted the directory, the S3 locally, as so it's then mirrored, and then I think I was able to then use the standard Linux tools, like if I wanted to do a find on the structure, that I could then apply that. Does that mesh with what your understanding?
Well, that means that your S3 server made that available. So yeah, I mean it's not unheard of, but if you depend on it, so yeah, if you depend on it, then someone out there could have S3 that wouldn't provide that, right?
Okay, fair enough. All right, thank you.
Yeah, no worries. Okay, so let's. Okay, we're going to switch to the other notebook now, where we talk about how to generate ZAR data. It's called conversion. Okay. Background again is at the top. So I mentioned bioformats, this library to generate or to read, 150 different file formats. You can do that on the fly. So your code can just use bioformats. Alternatively, you can use bioformats to generate OME TIFF. And that's what we'd like to see people doing more and more. Since OME TIFF has the performance issues, Glencoe Software came up with a suite of tools basically to speed up the process of generating OME TIFF. So if you need an OME TIFF, this is really how you should do it. Because ZAR and N5 are performant formats, multi-chunk, you can write them in parallel. They chose to use those as an intermediate data format. So bioformats to raw generates ZAR and N5, and then you can take the ZAR and N5 to generate the OME TIFF. So as a byproduct of their needing to create, we can all wave. As a byproduct of their needing to generate OME TIFF, we now have a fast converter to ZAR. So if you have the conda environment active, sorry there. Okay, I was just checking the chat. Then you have the bioformats to raw command. You can run help on it and get all the possible options. Many of the options are there for speeding things up yet more. You can say how many threads, what size the tiles should be. I won't go into any of that, but as if you have a couple 100 terabytes that you need to convert, then it starts to make sense to try to speed up each individual conversion. So we're only going to really look at two of the options. The first is that it's necessary to say that you want ZAR. By default, N5 will be generated because it's more native to Java.
George?
Yes?
Just short question. So this bioformats to raw, so within Java or within Python, does that also exist or not as easily?
So you can, I mean, bioformats to raw is written in Java, so you can use it from your Java code. If you were working from Python, then I would probably point you to Jamie from AICS Image.io, and I'll let him talk about that if he wants. Can you write OME ZAR yet?
We are just in the middle of releasing that. Like, yeah, I think Jackson's working on it right now. So I not right now. So as of the moment, no, but before the end of the year, you should be able to. Yeah. That's
Oh, I thought you were going to say before the end of the day. Yeah. So.
Oh yeah. Well, that's always possible with Jackson, but yeah, we'll see. It's definitely in the next couple weeks.
So for all intents and purposes, AICS Image.io, which I always find hard to say, is probably the closest parallel to bioformats in Python. So if anyone wants to do these types of conversions, I would start there. Yeah, full stop.
Also, if you have something that would be useful for us, just sorry to mention to throw this in here, but like if you work on an image format that is not like, so we currently are basically focusing on image formats that we use internally, like Zeiss, CZI, 3i, etc. But we're pondering like adding image format support for Olympus or something like that. But if there is a scope that you are particularly wed to, if you could put in a request on the issues for that support, that would be awesome. Just so we have some concept of what would be terribly useful for outside users. Anyway, thank you.
So a PSA from Allen Institute. Yeah, don't worry. I have warned him that he doesn't want to get in the business of supporting all the formats. So we're all on board with the same thing. Okay. So the other option that we need to add is the dimension order. The default in bioformats to raw is more for digital pathology. I think it's the C at the end version, and we want T at the end version, right? So basically, and this is time for all of you to then actually run bioformats to raw. I assume you're running the cell. It has a hard-coded name of GIF-ZAR at the moment. You can change the name if you want. The input is a little GIF from Tischer, which we have I'm skipping a step to actually generate the GIF. So you can run that cell. It will create in your local directory. So I assume in my repository, two things. One is metadata OME XML. So this is all the metadata that bioformats can parse from your input image. So obviously nothing from a GIF. But yeah, if you have a more complicated microscopy format, you will have all that metadata in that file. That may be useful to you. So for your own purposes, the developer of ViZAR and Viv, Trevor Mance, and colleagues, they make use of that in their GUIs. You know, they read that for metadata that they're expecting. Ultimately, the process of specification is we want this file to go away, right? We want to take all the metadata that exists in OME XML, and it should live somewhere in the ZAR. That's the goal. Then you have, this is a bit odd, a directory called data ZAR. So bioformats to raw just always generates, sorry, I used to be a kindergarten teacher, so I can't help like not waving.
I'm sorry. I will be less distracted.
Don't apologize. This is great.
Actually, just to go back on the command above, I was having issues making that work on my local system. It was refusing to store in the very directory where it was living, and when I added overwrite to the options, it just killed everything. So might be the mixture of versions that I'm having here. I just installed before the workshop, but never mind. It's just if someone else runs into that problem.
Okay, good to know. You would have to give me the, like, show me the output, but we can work on it. Yeah, we definitely don't want it to clobber things. So this kind of backs up a second to how bioformats works. Bioformats, actually the OME standard, lets you have multiple images in any file set. Sorry, I'm getting pinged on Slack. That means, so let's say you have a CZI file or something, or even a TIFF, that can have many individual multi-dimensional images in it. In bioformats world, those are called series, right? So you can have in one file set, you know, tens of thousands of images if necessary. So to work around that, bioformats to raw currently always creates a top level directory called data_zar. And then underneath that will give you your series. So we could have had, you know, GIF 0, 1, 2, 3, 4, 5, but our GIF currently just has a single series in it. So if we look into that file, into that directory, we can see all the bits and bobs that Will was showing us earlier. It's a tiny little GIF, so there aren't solutions. It's just the full scale data. You have the metadata, and then you have all of the chunks down a directory deeper, right? Okay, so here's where everyone needs to run something again, and you need to update what it's called, because otherwise it's going to clobber. So I'm going to upload the GIF to Josh. You can also put it somewhere deeper if you want. Foobar, that's up to you. Important is recursive. I'm pretty sure MC does a good job of telling you if you give it a directory that you must pass recursive. And then the upload, I mean, it's a GIF, so it's super fast, but it does work in parallel. You don't need to, for mid-sized data up into, you know, tens, maybe hundreds of gigabytes, I think MC will work fine. If you start to get in terabytes, then you might get into the ranges of issues that Tischer and I were having transferring from Germany to England. And then maybe you want to do something smarter. So yeah, as always, benchmark. And if you have problems, work around them, right? Okay, so at this point, you should all have data up on S3. Here, I'll check that everyone's doing their homework, and I'll look how much stuff we have in the bucket. Data_zar, that's not what I want to do. ls play i2k. Okay, so about 6 people did something. Then there are tools in MC so you can get the data back. You don't have to copy everything down. You can just concatenate it, cat it to standard out. So I'll look at mine. So that's the multi-scale metadata to say that this is actually an image. And at this point, so I need to update this URL. We're going to make use of Visar from Trevor. I'm going to change my ZAR URL to Josh. And again, with no server code anywhere, it's someone else's browser, static HTML and static S3, I can view my little GIF. So it's I2K 2020. Visar really needs a time player, Will, so that we can just like click play and go through the movie, but we'll get to that later. Actually, those are the Z slices, right, Tischer?
No, it's time.
It's time. Yeah, we need a movie player. Visar Netflix. Okay, so all of you who actually played along have now published something to S3, and you can view it in JavaScript. For people who do have Omero or know about Omero, there are also other tools for exporting directly from Omero to OMEZAR. So the tool, one tool is called Omero CLI ZAR. There are others, and we're happy to obviously talk through more of those. An important concept with all of this, let me bring up one slide that's already in Slack from earlier today. Someone asked the question, he was trying to get his head around all the pieces we just talked about. So an important concept here is that regardless of which of these tools you're using, the representation is completely isomorphic, right? So what we just did was we took a GIF and we generated a ZAR, then we uploaded the ZAR. It's the same format. You can access the ZAR remotely via Dask. It's the same format. You can download the whole ZAR. It's the same format. It never changes what you need to do. And what that also means is you can put a microservice in front of your existing storage. And when you talk to this thing, I think I have a working URL, it looks like it's a ZAR. So that's your business. You can do this on the fly if you do have a server somewhere, but at least the format then is well defined. Does that, oh, that's not what I wanted. So let's take, I have it over here. I already opened it. Sorry, I'm a monster when it comes to browser tabs. Okay, so what this is saying, so this is just some Omero that's running somewhere. And it's this microservice, and it just looks like it's a ZAR. So you could click, let me give you this link, sorry. So the important, Nicholas, I don't know why you're having problems. What you typed is certainly correct. We can get to that in just a second. So this is an Omero with a microservice that acts like your data is a ZAR. So someone can, pass this URL. Actually, I guess can we do that? Let's do it. Oh, wow, this is exciting. So we can pass this to vZAR. I didn't test this beforehand, and let's just see if it works. I don't even know what image this is. Someone want to do a drum roll? It looks like it's pretty slow. We'll come back to, or we'll keep talking and see if it shows up. This is something Trevor's done a couple of times. He's written various other little servers, nothing to do with Omero, that make TIFFs look like OMEZAR, make HDF5 look like OMEZAR. You can, to some extent, what this is becoming is a, call it the API or the protocol for all of us to access the data, so that, it's the thing in the middle as opposed to bioformats that we're using to access our data. And that's kind of exciting. Even if it's not showing up on screen. Okay, I'm going to shut up and see if anyone wants to say anything, ask any questions. Oh no, it's cores. Web browsers. So if you don't know, cores is something, every time you want to make data like this public, so put it on a little note somewhere. If JavaScript is giving you failures, then you probably need to talk to someone to enable cores. And all that, I mean, it's basically preventing people from stealing your credentials, but in this particular case, it's not a problem. It's public data. Please just let me show it. So sorry about that. But you can click through the URL I gave you and you can see all the metadata. There's actually a little more metadata here. You can see all the chunks, et cetera, et cetera. So have fun. Okay, I'm being quiet and drinking something.
Sorry, in the party.
Yeah, someone want to try it in Pari? You have the URL, do whatever you want with it.
Can I ask a format question? Or I guess it's really just a chunking question.
You can ask me a question. You can always ask me a format question.
So I guess for the OME, the new OME standard, is it opinionated in terms of how chunking should be done? Or so for example, like I noticed it looks like it's sort of each tile, like 2D tile is in a chunk, but if there was a reason we wanted all the color channels to be in one chunk together, is that okay? Or is that kind of?
Yeah, so the spec itself is agnostic. So that's, that lives at the ZAR layer, you know, it lives down low. There may be an assumption, a performance assumption in some application. Thank you, Will. You know, in one of the 3 implementations we've showed you today, there may be a performance assumption that would be annoying. Ultimately, though, that we should get rid of that. So you should be able to specify that, again, the chunking and the compression and the sub-resolutions that make sense for you in your application.
Cool, yeah, that makes sense. Because yeah, that's all, I guess, taken care of by ZAR. And so.
Exactly.
Yeah. Awesome, thanks. Actually, a related question. I saw in the Slack a bit ago, there was something about maybe like a rechunking library or something. I didn't have a chance to chase it up, but has that been something you've found good tools for, or like smart ways, for example, if you've chunked it one way for analysis? And maybe
yeah, it's a good question. So thank you for bringing it up. So we started this in the previous session. So where the link came from last time, someone was asking, why doesn't bioformats to raw allow chunking? And it really like you to set the chunk size. I'll open an issue to enable that in bioformats to raw. There are 1 of 2 things that are happening. Either they made an assumption because they're primarily dealing with digital pathology, you know, so their use case doesn't need it, or their algorithm assumes a particular ordering, right? And just they've chosen the optimal chunking. I don't know. But for everyone's purposes, it happens a lot. So you should almost assume it to be not standard, but a common occurrence that your users will want different chunking sizes for your data. You know, if you really have a large, valuable data set, you know, if one person's watching a movie and the other person is, you know, doing some form of deep learning that doesn't take time into consideration, they're loading the data in a different direction. Their code will be faster if you rechunk the data and save another copy. Now that's pretty annoying in terms of storage, but it definitely happens. The most common way, or the most naive way that one would try to do rechunking, is just to use Dask and basically, you know, open the data set, the array in Dask, and go save it with a different chunking. Turns out that's horribly inefficient. I can send you the issue, Kevin, if you're interested. So the Pangeo guys were like, you know, it was 4 or 5 zeros times too long. You know, it was just astronomical. There's some, I don't know if it's exponential or polynomial, and it was just going crazy. And that's what rechunker is for. So it's, you know, those guys had a problem. We want to rechunk quickly. I think just like with bioformats to raw, because ultimately bioformats to raw is also a rechunker in a weird kind of way. It's at the cost of space, right? So you need to have space available to unchunk a large portion of the data, if not all of it, I'm not quite sure, and then redo it. So, you know, if you don't have 2 or 3x your data size, you're in trouble. Yeah, it's a interesting computational problem. You know, if anyone has suggestions, by all means, jump in. I think what we will do probably in our upcoming notebooks and our upcoming documentation is just tell people, use bioformats to raw for the basics and then use rechunker for the next step. It'll be interesting if we can teach everything to read and write directly from S3, then you don't need the storage locally. I don't know if all that works yet. So that's kind of an open question mark.
Cool, that's super helpful. Thank you.
Wow, we're under 3 hours, aren't we?
I don't think that's a problem. I don't know. I mean, I might have.
Yeah, I'm not complaining.
Maybe you can have a quick look at the chat message from Niklas.
Okay. Yeah, someone else talk.
I actually have a question. I haven't talked with Zollfeld about for N5 the reasons for this, but once you get down to the actual chunks, everything it's just like ZAR was the same. You've got dot for each of the dimensions. And does anyone know why that wasn't done with subdirectories? I know ultimately once you get to the object store, it doesn't matter, but I think definitely on file systems, there are all kinds of problems with the overhead of managing the metadata for a single directory with millions of chunks or files in it. And I believe even the object store stuff does, especially when you're doing these listings and things like that, you can sometimes slash is a special thing. And there are some efficiencies to be gained by actually nesting and distributing your things that way rather than having one sort of parent bucket that has millions of things in it. Is anybody aware of why that? Because both N5 and ZAR seem to a couple of high level qualify. You've got whatever data sets and you've got groupings, and then boom, put everything else under here.
So there are a couple of points to that story. So you're right. N5's default was subdirectories. ZAR's default was the dots. ZAR chose that default because they were cloud first. And so they figured, oh, it'll be fine. It was a big mistake. The next version of ZAR will default to directories for all the reasons you just said, because it's a huge pain in the butt. The reason that we're not using it with OME ZAR at the moment is because I could get the Java code to support the dots, but not the Python code to support the slashes. So and that's because, and this is a problem both for N5 and for ZAR, so they developed their specs, then they built libraries on top of their specs and developed logic in the libraries that never made it back down to the spec. The specs didn't evolve when they had these type of issues. They did everything in the libraries. And that fundamentally means that you then have to recode everything in every language for every combination, right? So I think that's one of the big lessons learned and why we're trying to design a common spec both for ZAR and N5 so that problem goes away, because it's been quite a nightmare. I would hope to be able to introduce the slashes in OME ZAR early next year. So that's kind of the time scale, because we have at least 2 data sets that are basically unusable. We've done the conversion, but I think one directory has 14 million objects in it, and it's
Oh, that's good. I'm glad to hear that it's a problem for others and you're working towards a solution. That's great.
Yeah, so completely understood.
Are there also plans to consider adding other types of annotations to the standard, like ROIs that are polygons or like coordinates?
You guys heard my answer to this earlier. You want to take it up? Can you do it?
Will, come on.
I think the answer is just yes. There's been some discussion of it already on ImageSC. Well-known text was proposed as a potential solution. To get the details.
Okay, I'll look for those threads. Thanks.
Yes, certainly the OM. You should be able to find everything on the OME NGF tag on ImageSC. Yeah. Okay, sorry, Nicholas. I will fully admit to not even having tested Windows. I'm sorry. I can help you use Docker if you want.
It's no problem. I'll find the solution maybe.
All right. Let us know though.
Yeah. Can I? So are we already in the all questions asking series, or is this something to come?
No, I think we are in all questions. Yes. Yeah.
So I wondered. So you have the labels incorporated in your file already, and I was wondering if it's possible to put in different labels, like when you want to share your the results of different machine learning models, you could, or is there some possibility to incorporate like the different segmentations?
Do you have an example, Will? So you definitely can. And in fact, in some cases you have to. So I don't know if I have the ID in my head from our sample data, but when you have, for example, overlapping segmentations, then you want to separate that into multiple segmented images. Like we have one data set that has the cells and the nuclei both segmented. So you put those in two label sets because otherwise it makes no sense. You know, it just makes no sense. So yeah, so you can have as many labels as you want. One thing we want to add is metadata on where the segmentation came from. You know, who ran it, when was it run, what was the workflow. You start to get into kind of scary issues, and so we may end up just going put JSON here and then it's your business. But it's better than nothing, right?
Okay, thanks. And I have another question. Is when you have different positions or different replicas, you put them into the groups. Did I understand this correct?
Yeah, so we're kind of learning as we go. So labels currently use a different style. So there's a single group called labels, and you can put as many labels in there as you need, you know, so you can fill it up. So that's how you register labels on the image at the moment. But most likely in the upcoming update, in general, we'll probably have more and more examples of, let's say you've written a labels object and you want to say this label object should be applied to this image. So basically what Tischer was showing you in Groovy code, right? So when you want to make that kind of linkage, it will probably become metadata on one of the two objects. What people are asking for more and more is, can you also have that linkage go to another file? You know, so I think that will become kind of the standard metaphor that if you want to write two things, two arrays or sets of arrays that relate to one another, put them wherever you want, even in the same czar or different czar or a different server, and put the link, you know, write down the linkage on at least one of the objects and maybe on both. Does that make sense?
A little bit, but so I was thinking about so without labeling, just if you have one experiment where you have different replicas, or if you, so we are imaging biofilms and we image one biofilm, but and another, and they have in one biofilm, there are the three spatial dimensions, channel dimensions, and time dimensions. And how would I incorporate into one SAR file the other biofilms? This would be by grouping them on the higher level. This was more the question.
Yeah. So at the moment, so it's roughly the same answer. So in one ZAR file set, you could at the top put 3 groups, and each of the groups would be one biofilm, right? So that you could do today. The only thing you would need to do at the moment is when you give someone the URL to the biofilm, or if you want to tell someone, look at one of the biofilms, you would have to give them the full URL with the subgroup. So maybe this is the segue, Will, into plates. More and more, there will be metadata that describes a collection of images. So far, we've only talked about high content. So we've only written the specification for high content screening. So you can point at a group that says, here are the 384 wells of a plate. So it gives you a higher level object that you could use to talk to, to share with someone. There will be more of those collections. So that would be a question to you. Does it make sense to have, is there any metadata you want for the whole collection of images? Or are they really just 3 separate things and they have no relationship to one another? That's fine too.
Okay, thanks.
Here's an odd question. With ZAR, right? Like, let's say I have a ZAR file and there's a segmentation in there as a label. And then, one of our imaging scientists decides that, oh no, I've updated my segmentation. I have a new shiny version. Is it a terrible idea to update just that label object or file within the object, or does one need to regenerate the entire ZAR quote unquote file?
I want to say not my problem.
Yeah, okay.
So I don't get to decide. That's your business.
Okay.
Can I try and answer, Josh? So the way I understood it was that I think this is all directories and you can have many labels, label images. I thought you just copy the second version into the ZAR.
Okay.
That's what I would do, but is that correct, Josh? Yeah.
So that's the only feature that the spec provides to you. So the spec allows you to write multiple if that's what you want to do. Our librarian, so I'm thinking about the IDR, so when we get a data submission, the librarians don't want us to do what you're talking about, right? They would want us to copy the entire data to an annotation.
Okay, but if I have
primary
local,
and the label.
Okay. But okay, I see. But if it's my object store and we have, let's say, a first version of the segmentation was not so good, then we add the second one into the same ZAR and the third and that's good practice, or?
Yeah.
Yeah, segmentation is also just some kind of metadata on top of the pixel images. And I personally consider it very important and a big failure in, let's say, the DICOM standard that there is no, at least when I checked, that's a long time ago when I checked last time, there was no versioning of metadata. So you were not even, DICOM specifies that you must not change the metadata, which sometimes imagine DICOM as the medical standard that is basically produced by anything you have in an emergency room or whatever. So, but emergency room means you don't have time to do anything. So you just put in data and images and there is no metadata in there. So you are not allowed to change that stuff later on, even though they didn't have time to put in the patient's name and it would be valuable to have it in there. Personally, I would prefer much if you could just add it and have a versioning where you could go back to that.
Yeah.
It is super complex, I know. This is nothing I want to put any burden onto anyone here in the specifications. But let's go back to that segmentation or annotation example. Having, let's say, the first segmentation and you're not happy with it and update it there. and like knowing later on from the metadata of the whole thing that this is like the newer version of the other thing, which is the older version, which kind of represents the same concept, is something that I personally would consider very useful.
In that vein, so something we've been doing with our files that for TIFFs anyway, OMI TIFFs, is that we use Quilt, which does essentially data versioning. So if you have a file, like if just a small portion or like if a file within a folder structure changes, it essentially just, you get a new version. It doesn't, it essentially does the same thing that GitHub does, you know, in the versioning. So I don't know if that's compatible with the ZAR structure as well. I haven't looked into that, but that might be the kind of thing that would be nice for that, or something that could be still, I mean, Quilt's open source, so anyway.
Typing Quilt into Google is a bit difficult. What could I use as reference?
Okay.
I think something like Quilt data.
Okay, yeah, thanks.
So, Jamie, I mean, but Quilt, does it give you a S3 endpoint?
It is an S3 bucket. Yeah, like our stores on our Quilt sources are S3 buckets. I'm not sure.
Yeah, but what does the data look like? Like, so let me, you said you don't know what it looks like with ZAR. I don't think anything currently provides versioning of S3. I don't know of a way to version S3 objects so that you have basically the full directory, then you have the full directory again, and you can see the diff, right? Without using duplicate storage. I would love to find out if there's a solution.
Okay, let me just ping. Jackson actually works as a dev on their group sometimes, so I'll ask him.
Okay. So to clarify what I was saying for everyone's benefit, you know, what I've never heard of is a way for you to update your metadata without needing to duplicate your binary data. You know, so if Jamie's got that, then, you know, he gets a beer or whatever.
Okay.
Or we can do what CZI did and send cheesecake, right?
The Z stands for Zuckerberg or Zeiss?
Zuckerberg.
Okay.
But maybe just to make sure that what I said, that's possible. Or so I have this SAR thing, I have one raw data image in there, I have one label image, I can a month later just copy a second label image into it. Or and okay, I would have to update probably somewhere one of these .group or .attribute files. So is that A, possible, B, what you would recommend?
So A, possible, B, it's the best you can do at the moment. There is a downside, and it's a downside I didn't mention in the notebook. So let's cover it real quick, which is S3 is eventually consistent, right? Meaning if two people start writing the same data, so if two people start adding a label to an existing image at the same time, there can be a race condition and you won't know necessarily who won. There are ways to do it. There are ways to code for, please tell me if you haven't changed, but I certainly don't know of any of our code that does it. And I think it's less typical. So we should probably keep that in mind when we write the spec. You know, can someone add a label without clobbering someone else's data? And currently that's not the case. I mean, for the IDR data, I do it. You know, when we have an update, I just S3 upload because I know I'm the only person doing it. Thanks, Blair.
But Josh, this is only, it's only this one .set groups file, or that's the problem?
Yes.
Okay. Because all the other things have different, well, it depends if, yeah, no, okay.
Yeah, as long as you name things differently. So if you if you have your segmentation names based on user and timestamp, then it's probably okay. But if everyone tries to use the name masks, yeah, you've got a problem. And I mean, these are all things that a data management server would take care of for you. And now with S3, they're all client problems. So that's why public static read-only data is much easier. And it's kind of what I assume at the moment. And for everything else, we're going to have to figure it out, right? It may make sense, for example, for those of you who know Omero, to teach Omero how to manage S3 for you. And you don't try to do it yourself. You don't try to do it on the client. And Omero learns how to write to S3, for example. It's tricky.
Can I give this as information to the consortium of our next big grant discussing about how to store the data?
You're currently writing, or you want to write a big grant to do this?
No, it's actually, they're planning on, the grant has been assigned and they're planning on how to realize it. So, I mean, the money is there.
Okay,
so
sure.
They're okay, Omero with the S3.
Mm-hmm. So that's basically the very rough outline of the vision for Omero 6.
Mm. Yeah. Cool, amazing.
We'll see about that.
So Josh, now that S3 is strongly consistent, has that changed anything for you?
I haven't read that. Let me see what it says.
So
well,
and
also remember, every S3 is different. So if you're using Amazon and you're paying for strong consistency, then maybe. But I mean, I'll read it and we can discuss. Let me see if I can see quickly whether.
Someone posted this on one of our other Slack channels just yesterday.
But I think it'll depend if it's on a per object basis, because then if you have a process that's writing 1 million objects, what happens in the middle, right? I mean, it's fundamentally the transaction problem. If you try to have a large transaction, eventually it's going to slow things down. So you get rid of the transaction and then you don't have ACID constraints, right? So I don't think there's a good answer. You have to choose between performance and safety.
Mm-hmm.
Sorry for nerding out.
Okay, so just with Quilt, if you have a Quilt repo and you change a text file within it, it will do a diff on the entire thing, but it will only essentially it's only pushing the changed file. So if it's just the text file that changes, it only pushes the text file into the new commit. So I think so. It is doing essentially the equivalent of what Git does or GitHub.
But on the server, you only have one version at a time.
Well, I think it's like I think it's treating the entire thing, your Quilt store or your Quilt object, as a repo essentially. And then so if your text file within it, it's like a text file within your GitHub repo changing. So the commit would, if you check it out, it'll pull out everything, but the version will only be stored when you make a change, it's just changing the one piece that changes. Does that make sense?
That makes sense, but I still think so. I guess what I would need is a URL to the previous version. That's what I'm looking for.
Oh yeah, so the commits are, you can pull out the previous commit. Yeah.
But an S3 URL to a previous version exists?
Well, okay. When you check out, so this is all a Python interface. So I'm not sure how the URL interface, like how you could maybe bend that or not, but that's that aspect of it. Also, this is probably not coming anytime soon, but there is like, because of their, but it is apparently like Google Cloud support is on their roadmap eventually for Quilt, but that's a big project. So anyway.
Mm-hmm. I feel like there are people who have been pushed in the background who should probably be asking what they would like to see happen as well. So, I mean, so for everyone's benefit, there's a monthly NGFF call for the developer type, so they can always find me when they need me. But any users suffering in the background or just tired of listening to us? And I'm getting a private motion in Slack to adjourn.
Just in this little demo for Quilt, it looks like it does have a separate URL for each version.
Mm-hmm.
Here, I'll send you the example I'm looking at.
Cheers. Anyone who's hiding? Hopefully this has been useful. Okay. Got at least a few thumbs up. Look forward to seeing lots of data in the new format, but let us know how it goes. All right. Take care, everyone.
Thanks, guys.
Thank you.
Yeah. Thanks.
Thanks for all the great work, guys. Nice workshop.
Thanks for listening so long.
Cool. Thanks.
Yeah. Gladly. Bye-bye.